today we're going to talk about certain and uncertain digits. Welcome to lesson 1.4. Certain digits are numbers that are going to remain the same regardless of who measures them. We're going to look at that here in just a second. Uncertain digits are digits that can be estimated and will vary. When you report a measurement, you must include all the certain digits plus one uncertain digit. So let's look at this burette. When you read volume in the laboratory, you read at what we call the bottom of the meniscus. So water, because of its cohesive properties, will form a meniscus, so the sides will be higher than the middle. And in this particular inset, we need to look at first how this is measured. So if we look, this is a burette, reads backwards, so our measurement's going to be somewhere between 20 and 21, and it looks like there are 10 increments between 20 and 21. So this, but for sure, everyone can say this is at least 20.1. Okay, so those are our certain digits, 20.1. We are going to guesstimate the next digit, and that's going to be our uncertain digit. So this tells us that the volume of liquid in there is at 20.15 milliliters. If you're working in a lab group, someone in your lab group may have argued with you and said, no, I think it looks like 20.14. Or someone else might have said, I think it looks like 20.16. All of those are correct because certain digits measured the same by everybody. Uncertain digit is based on the individual looking at the measurement. A measurement will actually always have a degree of uncertainty, and that's going to depend upon the precision of the instrument. Now, please keep in mind that when you're looking at a digital device, like a balance, maybe a thermometer, those devices the data on those, the reading on those, the measurement on those must be recorded as all of the digits it shows. When we're talking about the uncertain digits, we're talking about what we call an analog device, which has a scale that you need to use. Okay? So any measurement always has that degree of uncertainty. We actually use significant figures, and the uncertainty is going to be found in the last digit, and it's always assumed to be plus or minus 1 unless otherwise noted. So on that last example, I said 20.15, I said 20.14, or 20.16 is acceptable because that's plus or minus 1 from that uncertain digit. So here's an example. In analyzing a sample of polluted water, a chemist measured out 25.00 mils of water with a pipette. So they're looking at that volumetric pipette that we looked at earlier. At another point, the chemist used a grad cylinder to measure 25 mils of a solution. What's the difference between those measurements? Well, on this one, you know for certain these three digits, and then this one is our uncertain digit. With this one, we know if this is written according to our rules, we know for this is our certain digit, the last digit reported those are uncertain. So this one could be anywhere from 25.01 to 24.99. This one could be anywhere from 24 to 26. So very big difference in the type of measuring devices that we use. There is a difference between accuracy and precision. A lot of times students will use these interchangeably. If you're going to look at how accurate a measurement is, you have to know what the true value of that measurement is. And so accuracy is closeness to the true value. Precision is having measurements that are very tightly spaced together. Almost every chemistry book I've ever seen uses a dartboard to show these. And so in example A, these are neither accurate nor precise because accuracy would be hitting the bullseye. Precision would mean they are close together. So these are not accurate nor are they precise. In this bullseye, in B, they are not accurate because they're not anywhere close to the bullseye, but they are precise because they're fairly close together. And then in C, we have both accuracy and precision because they are very close to the bullseye and all of the measurements are close together. You can't have, if your measurements are not accurate, You can have some different types of errors. So you could have a random error. That means that the measurement has an equal probability of being low or high. And sometimes that just occurs because of estimating that last value. So random error is just something that happens in lab. We all take measurements, random error happens. Systematic error means it's always in the same direction. So that would be like me having a scale at home that I knew was 5 pounds off. And so let's say it read 5 pounds low. That means every time I get on that scale, it's not going to be the accurate value, but it's always going to be low. And so systematic errors are usually caused by your equipment and perhaps your equipment not being calibrated. The bullseye on the far left shows large random errors. The bullseye on the right, those are small random errors, but a large systematic error because it's not equal to the exact value. And then the last one shows, because they're not all exactly the same value, shows small random errors, but no systematic error because they are close to the value and not either all high or too low. A lot of times when we're looking at the work of a chemist, we kind of relate precision and accuracy. Precision is often used as an indication of accuracy. If you get several measurements that are close together, then you can assume that systematic errors are absent and that your analysis is good. So we're going to look at some data. We're going to check the accuracy of a graduated cylinder. What we're trying to do is, well, actually what we did is we filled the cylinder to the 25 mil mark using water, and then we actually put that in a burette and determined what volume was actually in that cylinder. So every time the grad cylinder showed 25 milliliters. When we put that volume in the burette, these are the values that we obtained. Is the grad cylinder accurate? Well, if you look at the difference in the average, that grad cylinder is not very accurate. Is the grad cylinder precise? These are fairly close together, so I would say the grad cylinder is precise, but it's systematically delivering the wrong volume. So that's telling you the student has good technique, but the value is not close to the true value. So the student has good technique, the device itself is not accurate. This is a burette. We put it to the 0 mark with a solution, and then we transfer solution to the beaker. So basically it started out at 0.00. What volume of transferred solution should be reported? So notice between 20 and 30 there are 10 marks. So every milliliter is marked. So we can say for certainty it's at least 22, and then we can guess one more. So we would say use answer choice C because these are our certain digits. This is our uncertain digit that we guessed. Another example here. The boiling point of a liquid was measured in lab with the following results. So we measured it 3 different times. The actual boiling point is 28.7. Okay. The results of that determination, they were close together, very close together, but they weren't anywhere close to that. So they were precise but not accurate. So good chemist, bad thermometer. So they're precise but inaccurate. So the answer choice would be B. Just a couple of definition questions to finish out. Blank reflects the reproducibility of a given type of measure. So if you're trying to get the same value each time, that's a measure of precision. So the answer choice on that one is B. And then the last slide is the blank is the agreement of a particular value with the true value. So if you're trying to look at how your value compares to the true value, you're looking at accuracy. Okay, well that concludes lesson 1-4. We're going to talk more about significant figures and measurements in lesson 1-5.